right, so we're going to run through cell phys. Again, most of it is review, but I just want to make sure that we're all on the same page when we start, because we're going to spend a lot of time talking about this. So, if you remember from biology and undergraduate physiology, cells are made up of organelles. Different organelles have different functions, just like the organs in your body have different functions. So the Golgi apparatus here is responsible for trafficking. It's the traffic director inside the cell. Rough endoplasmic reticulum has ribosomes, so it's responsible for synthesizing proteins that need to be associated with a membrane, so things that you're going to secrete by exocytosis, or things that are going to be structural and are going to get embedded into the membrane, the cell membrane. Smooth endoplasmic reticulum does not have ribosomes, so it does not make proteins. It makes fatty acid molecules, so it'll make steroid hormones, any other types of fatty substances. Centrioles are there for cell division. They provide the anchor for the cytoskeleton that we will talk about. And of course, you all know there's other things like lysosomes for digesting, peroxisomes for detoxifying, mitochondria for making ATP, etc. Protein synthesis pathway starts in the nucleus. Genes. So gene transcription. Genes are transcribed into messenger RNA. Messenger RNA leaves the nucleus, goes out to find a ribosome, where it can be then translated into a protein. So protein synthesis is the way by which old proteins, worn out proteins, are replaced by the cell. Also, how cells can change their component receptors on the membrane. It deals directly with pharmacy, because one of the things you may see with people who are taking a particular medication is over a period of time they start to get used to that dosage and you have to up the dosage. And that's because the cells that are binding that are trying to overcome that overstimulation of the drug by decreasing the number of receptors available. It's called receptor downregulation or desensitization. And of course, any cell type that secretes things for a living, neurons that secrete neurotransmitters, endocrine cells that secrete hormones, goblet cells that make and secrete mucus. These are all going to require all of the organelles necessary for this protein synthesis pathway. Right, so cells that have as their function secreting things are going to have a lot of rough endoplasmic reticulum, a lot of Golgi apparatus, because they're going to be doing a lot of this translation and packaging of materials for secretion. Proteins you will find on the membrane. This is where we're going to spend most of our time as pharmacists. Drugs that you are going to give, for the most part, are going to be binding to receptors on the cell membrane. Some of them may be lipid soluble and act inside the cell, but most of them are not. So you've got different types of proteins. Proteins can form channels, allowing for ions and other substances to move into and out of the cell. They can be transport proteins. They can be signal proteins. They can just be proteins that are associated with a particular cell type that when immune system cells stop by to visit, they read those proteins and say, well, that belongs here. It's not a foreign invader, so I'll leave it alone. And lots of other functions. Integral proteins are embedded in the membrane. If they go through both the inner and outer membranes, they're called transmembrane proteins. If they're associated with either just the outer layer or just the inner layer and do not embed themselves into the membrane, then they are peripheral proteins. Cells maintain their shape and transport things within the cell from one point to another using the cytoskeleton. It's basically the intracellular railway system, and also provides your structure. So a simple cuboidal cell is cuboidal because the cytoskeleton inside of it is making it cuboidal. It's a white blood cell and it's amorphous. It doesn't have any particular shape. Its cytoskeleton is constantly changing. There is no set arrangement. So the cytoskeleton gives the cell its structural integrity, and it determines the shape of the cell. Also provides a nice network inside the cell to transport things around. Made up of 3 components. Microtubules are the largest. They are there for structural support. They give the cytoskeleton its strength. The smallest are microfilaments. They're also the most numerous. Those are there for primarily transportation and connecting things together. And then intermediate filaments are not as big as microtubules, not as small as microfilaments. And they exist for a variety of functions. Holding organelles in place, things like that. Cells need to be knitted together to form a tissue. And the way they are knitted together is via junctions. There are different types of junctions that exist between cells. Junctions, basically, you're not connecting the membrane of one cell to the membrane of another cell. Because if you do that, the membranes will fuse together and you will have one big cell. Right? Cell membranes are just like bubbles. You put them together, they fuse. So the membranes of adjacent cells never touch because they don't want to fuse together. They want to maintain their individuality. So what these junctions essentially are proteins that extend out. They're usually connected to the cytoskeleton underneath the membrane. They extend through the membrane out into the extracellular environment and come into contact with proteins found on adjacent cells. Now, tight junctions are found in places where you want to make the membrane or the tissue relatively waterproof, watertight, so that you don't get a lot of fluid traveling from the apical portion of the cell in between cells to this basolateral portion. Right, so it prevents any fluid that's here from traveling between the cells and ending up here. So it keeps tissues in something of a watertight state. It's not 100% effective. There's always some leakage. But you'll find these tight junctions in places that are associated with a mucosal membrane, right? Because you don't want water leaking across membranes. Desmosomes are interesting. If you know what a rivet is, they're basically rivets. They basically help to hold cells together. Because they're circular and they're not tightly knit together like these tight junctions are, they don't prevent any transport in this paracellular pathway, but they do hold adjacent cells together. There are also hemidesmosomes, which are similar but they're usually associated with attaching the epithelial cells to the underlying connective tissue. And then, of course, gap junctions. We're going to talk about gap junctions when we deal with smooth muscle and cardiac muscle, because gap junctions allow for ions to travel from the cytoplasm of one cell to the cytoplasm of another cell. So if one cell depolarizes or becomes charged, all the cells that it's connected to via gap junctions will also become depolarized and charged because they share cytoplasm. And here you see structure for gap junctions. We'll find them in cardiac muscle and smooth muscle, and we'll talk about them to some degree in neuronal glial cells. Basically what they are proteins made up from something called a connexin. And they're just channels in the membrane of one cell that attach to the channels in the membrane of another cell and connect them together so that they can transmit an electrical impulse faster than you would find in any other system. Gap junctions are associated with what is called an electrical synapse. All of the synapses that you've probably talked about so far have been chemical synapses, where one cell, like a neuron, releases a neurotransmitter, binds to a receptor on a postsynaptic cell, on a target cell. Electrical synapses, basically depolarization or action potential spread from one cell to the next via these gap junctions. Much faster mode of transportation, much faster way of transmitting a signal, but it's limited because it's only an electrical signal. Chemical signals give you a variety of responses. Electrical signals give you one response. That's it. We talked about protein synthesis. Protein processing occurs in the cytoplasm. We'll talk about this in the endocrine system mostly, somewhat in the nervous system, whereby products of a gene can be processed into multiple products by post-translational modification and by rearranging gene segments or mRNA segments, etc. One of the things that happens inside of rough endoplasmic reticulum is modification such as adding or taking away chemical groups from what was just made via protein synthesis. So methylating something, adding a methyl group to it, will do one thing to the chemistry of the molecule. If you phosphorylate it, it'll do another thing to the chemistry of the molecule. So these modifications whereby you add or tag these proteins can occur in the endoplasmic reticulum. And then transport, things are prepared for transport in the Golgi. So the Golgi apparatus is going to tell what was made. It's going to put a signal or target molecule on the vesicle that comes off of the Golgi apparatus to tell it where to go. Either it's going to go to the membrane for exocytosis, it's going to go close to the membrane and dock on the cytoskeleton and wait for the signal for exocytosis. It's going to be a digestive enzyme that's going to be part of a lysosome. It'll be a peroxidase enzyme that'll be part of a peroxisome. All of these different possibilities. Now we've got the basic, what is called a constitutive secretion pathway. That's basically the default pathway. Constitutive secretion occurs all the time. It's basically just membrane recycling. What you try to do on this membrane is you match endocytosis and exocytosis so that the cell membrane doesn't get any bigger or any smaller. Right? Because if there's more exocytosis than endocytosis, then you're going to be putting more membrane on the cell, and it's going to get bigger. If there's more endocytosis than exocytosis, then you're going to be taking more membrane away from the cell membrane, and it's going to get smaller. So the constitutive secretion pathway is concerned with a few things, and one of them is to maintain that normal membrane size. So matching what is put into the membrane with what is taken out. So endocytosis, there are different pathways for that as well. We'll go through some of those. You'll go through some of these also in immunology, if you're taking immuno now. Much of what is endocytosed ends up fusing with a lysosome so that the digestive enzymes in the lysosome can break down whatever came in via endocytosis. There may be some recycling, or there may be some other pathways, but for the most part, this is what occurs. The other type of exocytosis is regulated secretion, different types of regulated secretion that we'll talk about. One is, it depends upon the target that the Golgi puts on the vesicle. So some of the vesicles that are coming off the Golgi will be digestive lysosomes. So they're going to have a different signal molecule. Some are going to be carrying neurotransmitters or hormones. They're going to be attached to the cytoskeleton until the signal for exocytosis comes. So they're going to have a different signal. And then things that are going to be secreted, they're going to have a different signal. So the Golgi apparatus uses different signals to tell different vesicles where to go in the cell. We got that. Sometimes that's confusing. All right. So there's a drug called Brifildin A, which is primarily used in research. It's not usually given to people, so you may not be familiar with it. It blocks vesicular transport from the endoplasmic reticulum to the Golgi. So what effect would you expect this to have on a cell's ability to release a neurotransmitter or a hormone? Yeah. If you decrease vesicular transport in any part of that protein synthesis pathway, you're going to reduce the release of whatever the product was, whether it was a neurotransmitter or a hormone. It's referred to as bulk transport. Bulk because they're substances that are too big to travel through channels or facilitated diffusion carriers or anything like that. These are large substances that have to be moved across the membrane either by endocytosis or exocytosis. Bulk transport. Bulk transport occurs because of what's called a snare system. There's a snare associated with the vesicle. That's called a V-snare. And then there's a snare associated with the membrane, and that's called a T-snare. So vesicular snare, target snare. So for exocytosis to occur, these vesicles, which are normally stored attached to the cytoskeleton, the signal for release of those vesicles is calcium. So then these vesicles will detach from the cytoskeleton. They'll float or move towards the membrane. And when those snares come into contact with one another, they will begin the process of exocytosis. So those snares coming together allow for some peripheral proteins that are found on the inside of the cell to form a fusion complex. And so the cell membrane and the membrane of the vesicle fuse together, and whatever was inside can now enter the extracellular fluid. Now it works in the opposite direction for endocytosis as well. So some substance on or near the membrane triggers an endocytotic event, usually binding to a receptor. And so what will happen are, so V snares and T snares that were left over from a previous exocytosis event are found in the membrane. They will form the membrane fusion complex only in reverse. So then this will allow the vesicle to pinch off from the plasma membrane and form an endocytotic vesicle, and the snares will disassociate. Right, so it works in both directions. You've got endocytosis stuff coming in, exocytosis stuff coming out. Anything that is soluble, that dissolves in water, is going to be within a vesicle inside the cell. That way you can control where it goes. If it's not in a vesicle, it floats around wherever it wants to. Things that are not water soluble, things that are lipid soluble, they're a little more difficult. They usually have to be bound to proteins in order to keep them from just floating away anywhere. Signal transduction. What's transduction mean? Anybody remember? Yeah, sending a signal. It converts from one form to another. So changing. So one way of transduction that you're familiar with is you get an electrical signal that comes through your headphones, from your phone or your iPod or whatever, comes up to your earpiece, and then it causes a vibration of a membrane which produces sound. Right? So conversion of that electrical signal to a mechanical or physical signal that causes vibration is transduction. Transduction for cells is converting an extracellular message into an intracellular message, or the opposite. So it's basically converting a signal from one form to another. Signal transduction as far as across the membrane can involve ion channels. Ion channels opening, allowing for ions to diffuse across the membrane, either into or out of the cell. That's a form of signal. That's an electrical signal. Ion channels are referred to as ionotropic receptors if they are the types of receptors that need to bind a ligand in order to open. Metabotropic receptors are receptors that don't transport ions. They're receptors that change the metabolism of a cell. So metabotropic receptors are things like G-protein coupled receptors, GPCRs. Roughly 50% of the drugs that you prescribe bind to G-protein coupled receptors. So it's important that you understand how these guys work. And then there are receptor enzymes. So an extracellular signal molecule binds to the receptor. There's an enzyme inside the cell that's associated with that receptor that gets turned on or off when that binding occurs. You with me? Again, these are all. The thing about the boron medical physiology book is they have the best diagrams. So I like to use their diagrams a lot. The bad thing about the boron physiology book is if you look at it down on reserve, it's a tome. It is huge. It's like 1,200 pages or something. And it's written, the authors are British, so it uses the Queen's English. So some of the things, like they put an E on the end of neuron. There's different spellings for things, and it's written in a way that it's a little more difficult to understand. So I don't use the book, I just use the diagrams. So signal transduction. Over here we have ionotropic receptors, right? Neuromuscular junction, acetylcholine released from a skeletal muscle fiber, or excuse me, from a motor neuron, bind to acetylcholine receptors on skeletal muscle fibers, causing them to open sodium channels. All right, so that's an ionotropic response. The G protein coupled receptor response, the ligand in the extracellular fluid binds to the receptor. It stimulates, so normally these receptors, the inside, the G protein is trimeric. It has an alpha, beta, and gamma subunit. And it uses GTP as its energy source rather than ATP. That's why it's called a G protein. Because the enzyme associated with it is a GTPase. So binding of the ligand can cause dissociation so that the alpha portion can drift away from the beta and gamma subunits and then activate enzymes that are found in the membrane. The beta and gamma subunits stay together. They can drift away from the receptor as well, but they tend to alter opening and closing of channels in the membrane. So the catalytic subunit is the alpha. That's the one that has enzymatic activity. And the beta gamma are going to associate with other proteins in the membrane, cause different changes. And then receptor enzymes. So the ligand binds to the receptor on the extracellular surface of the cell. That causes the protein traveling through the membrane to change conformation. It'll somehow twist or move such that these enzymes that are associated with the cytoplasmic tail of the receptor become activated. There are different kinds of enzymes. The one that we're most familiar with is what's called a TRK, a TRK, tyrosine kinase. So those are the most common. There are also serine and threonine kinases. What's a kinase do? Yeah, phosphorylates. Right. Active transport. Sodium potassium ATPase, primary active transport. It's extremely important. Number one, it's necessary to maintain a normal resting membrane potential. Number two, it's necessary in order to keep all of these other secondary active transporters functioning. Because what the sodium potassium ATPase does is it directly uses ATP, cell energy, so it's primary. Pumps potassium into the cell to keep intracellular potassium levels high. Pumps sodium out of the cell to keep intracellular sodium levels low. Right? Those gradients of sodium being high on the outside, potassium being high on the inside, allow for you to use that charge separation as a means of kinetic energy. Right? Potential energy is when the charges are separated but they can't move. Kinetic energy is when the charges can move. That kinetic energy can be used to do some type of work. So secondary active transport is simply using the products of the sodium potassium ATPase, right, the sodium and potassium gradients, as a means to transport substances. So as sodium, which is high in the extracellular fluid and low in the intracellular fluid, diffuses into cells, that movement is kinetic energy. That can be coupled by this transporter with the movement of an amino acid or glucose. Right, so that's secondary active transport. The energy is coming from the sodium gradient that was created by primary active transport. Same with all these other transporters: sodium chloride, potassium chloride, sodium hydrogen exchanger. Right? So these are all examples of what is called symport, S-Y-M-P-O-R-T, because everything is moving in the same direction. Also called co-transport. And then there is antiport, A-N-T-I-P-O-R-T. Antiport means that the substances are moving in the opposite direction. So in this case, as potassium leaves the cell, it allows you to pump hydrogen ion into the cell in this exchange mechanism. So this is called a countertransporter or an anti-transporter or an exchanger. Quick show of hands. How many of you already knew all this stuff by the time we got here? Yeah, I know, but I have to go through it anyway. Cell-to-cell communication. We talked about gap junctions being an electrical synapse, whereby an electrical signal can rapidly be transported from one cell to another or through a group of cells. Synaptic transport, what we're going to be talking about with the nervous system. Basically releasing a neurotransmitter into a synapse, which is a gap in between two cells, binding to receptors, causing signal transduction on the postsynaptic cell or the target cell. Paracrine and autocrine. Autocrine is where a cell stimulates itself, so whatever molecule it releases binds to receptors that it has, causes self-stimulation. Or it might be a way of controlling how much of the substance is released. So as the substance outside the cell builds up, binding to autoreceptors may turn off release of that substance. So it's a way of stimulating the cell to maintain normal extracellular concentrations of these substances. Paracrine, there is release of a substance into the interstitial fluid, and neighboring cells that have the receptor for that substance will respond, as opposed to endocrine, where the substance is released into the plasma and travels everywhere in the body. Right, so paracrine is local, endocrine is everywhere. Calcium is an important signal, especially for exocytosis, but it's also an important signal for other things. It's kept in very low concentrations because in high enough concentrations it activates higher concentrations of enzymes that cause the cell to die, and that's usually bad. So we have different ways of calcium signaling. Voltage-gated calcium channels, so when the membrane potential changes, it may open voltage-gated calcium channels, so calcium can come into the cell. Ligand-gated calcium channels, some ligand binds to the extracellular surface, the receptor here, that opens a channel. Mechanically gated calcium channels, so when the cell is stretched or deformed or somehow changed, mechanically gated calcium channels can open. So that's the mechanism whereby if you stretch a muscle cell, a smooth muscle cell or a cardiac muscle cell, they will respond by contracting because mechanically gated calcium channels open, and that triggers a contractile response. Store-operated calcium channels made by Target. Not that kind of store. Store-operated calcium channels are there so that you can maintain normal calcium levels within the endoplasmic reticulum and within the mitochondria, the two places inside of cells where calcium is stored. So if calcium levels in storage, that's where the store-operated comes from. So if the calcium that's stored inside the cell begins to drop, then store-operated calcium channels open so that calcium can come in from the extracellular fluid and then transport it into the ER or the mitochondria. Free calcium does a variety of different things. Its primary thing that we're going to talk about is interaction with calcium-sensitive enzymes, enzymes that are turned on whenever they bind calcium, and calcium-binding proteins. Calcium does a number of things, as we already know from previous. It creates depolarization. When calcium channels are open, calcium comes into the cell becomes less negative, creates a depolarizing event. Generates action potentials in some types of cells, like cardiac muscle cells. Regulates enzyme activities, turns on, turns off different enzymes. We're going to talk primarily about calmodulin in smooth muscle cells. Mechanically gated channels for stimulating muscle contraction, and of course store-operated channels for making sure that the stores or the storage inside the cell is appropriate. Digitalis, foxglove, foxglove. It's a drug that blocks the action of the sodium-potassium ATPase. So if you decrease the action of the sodium-potassium ATPase, what effect would that have on calcium in the intracellular fluid? So take a quick peek at this, which we looked at before. Okay. Now, blocking the sodium-potassium ATPase would do what to calcium levels inside the cell? I see a rainbow of colors. So let's go back. So look at this. Right? The sodium-potassium ATPase is responsible for what? What's it doing? Maintaining memory potential. How?
Ion distribution.
Of what ions?
Potassium,
Sodium, potassium. So it creates the separation where sodium is higher on the outside of the cell and potassium is higher on the inside of the cell. Right? It also allows those chemical gradients, those ion gradients, to be used for various transport mechanisms, like this guy down here, the sodium-calcium exchanger. So you can get rid of calcium that's inside the cell by exchanging it for sodium that's outside the cell. And this works because sodium's higher in the extracellular fluid, lower in the intracellular fluid. So sodium will diffuse down its concentration gradient and then allow calcium to be transported outside of the cell. So blocking the sodium-potassium ATPase causes what to happen to calcium inside the cell? Yeah, that looks better. It's going to increase, right? Sodium is not being pumped out into the extracellular fluid as rapidly. So the gradient between sodium inside and outside the cell is going to decrease, decreasing your ability to get calcium out of the cell. That exchanger won't work as well if the sodium gradient is not high. Right? So if calcium stays inside the cell longer, the heart contracts more forcefully. So digitalis is given to people to give them more forceful heart contractions. G-protein coupled receptors. I've gone through this, and this is something we're going to go through in more detail. Comes in two forms. The IP3 DAG system, where a ligand binds to a receptor. The alpha subunit, the G protein, is called a G sub Q. So this alpha subunit associates with phospholipase C when it's released. Takes a membrane phospholipid called PIP2, converts it into diacylglycerol and IP3. IP3 is soluble, so it diffuses through the intracellular fluid to the endoplasmic reticulum, where there are IP3 receptors telling the endoplasmic reticulum to release calcium into the cytoplasm from storage. Diacylglycerol activates an enzyme, a peripheral protein found near the inside, the cytoplasmic face of the membrane, called protein kinase C. C stands for calcium, so it's a calcium-dependent protein kinase. So the combination of DAG activating the enzyme and calcium being present causes the enzyme to affect a number of metabolic activities of the cell. And that's what I just said. Cyclic AMP is the other second messenger system. There are two different G proteins. There's a stimulatory G protein, GS, and an inhibitory G protein, GI. So the alpha subunit for the GS protein activates cyclic AMP production by activating the enzyme adenylate cyclase. The inhibitory G protein, the GI, turns adenylate cyclase off, decreasing cyclic AMP activity. And then all cyclic molecules found in the intracellular fluid, whether it's cyclic GMP or cyclic AMP, are deactivated by phosphodiesterase enzymes. Phosphodiesterase enzymes take a cyclic molecule and make it linear. Once it's linear, it's inactive. And cyclic AMP activates a different protein kinase called cyclic AMP dependent protein kinase, or protein kinase A. And that's what I just went through there. Receptor enzymes, we'll go through them as we go through the organ systems. And then here's just an example of some various ligand effects from your book. Again, we're going to go through every single one of these as we go through that particular organ system. Any questions? All right, see you Wednesday. Don't forget to let me know your seating preference, por favor.